Hello, everyone, and welcome to today's webinar on Humanized Tauopathy and HUGO mouse model for AD. Thank you all for joining us. My name is Sydney, and I'm the International Product Marketing Manager here at Cyagen, and I will be your host for today's session. And we have a fantastic presentation lined up for you, led by our distinguished speaker, Dr. Maurice Cui. Hi, Maurice.
Hello, Sydney.
And Dr. Cui is the study director of the Cell and Gene Therapy Department at Cyagen, where he leads research in neuroscience and gene therapy. With over 5 years of neuroscience research and 2 years of clinical experience as a neurologist, he brings a unique blend of scientific and clinical insight to his work. He is highly skilled in using cell and animal models to study disease mechanisms and evaluate new therapies. So before we begin, a quick note, we encourage you to ask questions throughout the presentation, and please feel free to type your questions into the chat box at any time, and we will address it as many as possible during the Q&A session at the end. So without further ado, please join me in welcoming Dr. Cui, and let's get started.
Here regarding its very interesting mouse models. Today, as the title shows, I'm going to share with you about the humanized tauopathy in our HUGO mouse models and its indications for advanced AD preclinical studies. We are going to go through several topics, including the Cyagen HUGO mouse models and the details of humanized tau mouse models, and also several outlooks regarding how we're going to advance further our HUGO mouse model. First, we should talk about what is HUGO. HUGO is short for Humanized Genomic Ortholog. That means replace entire mouse gene by human patterned technology. We could acquire this kind of model from 6 months time frame. That is the fastest. And latest, I would say somewhere between 9 to 12 months. This kind of model is valuable because we think with a human gene inserted in a mouse, we could get a humanized process. That means humanized physiology and sometimes humanized pathology in the mouse model. With this kind of humanized process, we could be using this kind of mouse model for diagnosis of human diseases and also for test treatments for human diseases. As this is closely related to human, we call it translational relevance. And it can be used vastly in the research field we call translational medicine, where we use mouse models to study human disease. Until today, so far, we have at Cyagen over 100 HUGO strains, including all sorts of genes related to different human organism functions. Like in ophthalmology, we have humanized ABCA4, humanized rhodopsin, humanized VEGF-A, etc. As for the neural part, we have humanized tau, humanized MECP2, and humanized SNC, and humanized DMD, etc. In the immune system part, we also have the humanized PD-1, humanized CD47, etc. And in metabolism and cardiology, we have humanized GLP-1 receptor and humanized PCSK9, etc. As for today, we focus in the neurology part. More specifically, we focus on the humanized MAPT. Humanized MAPT or MAPT is short for microtubule-associated protein tau, or we can just call it tau. Tau is a skeletal protein which plays a key role in promoting microtubule formation and stability, and may also contribute in some developing and also sustaining the neuronal plasticity. As shown here, by comparing the amino acid of murine tau and human tau, we see some differences, we also see some similarities. To be more clear and easily clarified, we usually classify tau isoforms using two key markers. One is called the N-terminal inserts, but only in human we see 3 repeats. This fact matters because though we see many 4-repeat tau-associated diseases such as the PSP, CBD, and AGD, we also see 3-repeat tau exclusively associated diseases such as Pick's disease. And more frequently, we see the 3 and 4-repeat tau-associated diseases such as Alzheimer's disease, Down syndrome, primary age-related tauopathy, and other dominantly inherited dementia. That being said, if we only introduce human mutation in mouse, we could, maximally speaking, study 4-repeat tau-associated diseases because there are only 4 R-tau in mouse. With the entire murine MAPT gene replaced by human gene, we have the opportunity to study all those 3 and 4 R-repeat, tau-repeat associated diseases using our HUGO mouse models. To be more specifically and more appropriately study human MAPT-related diseases, in addition to replacing the murine MAPT gene by wild-type human MAPT gene, we also made another 2 mouse models carrying the P301L and P301S mutation, respectively. They are the most popular prevalently observed mutations in human patients with either the frontal lobar degeneration or the Alzheimer's disease, etc. Hence, we chose them. Since all those diseases features dementia syndrome, we validated our humanized MAPT mouse models, focusing on their learning and memory deficit. We designed our validation experiments like this. We acquired female and male mice, each 10, include to these experiments. And we validated their behavior alterations at 3 months old time point, 6 months old, and 9 months old. As shown here, at 3 months old, we started validating their motor functions and also other anxiety levels, basic behavior levels using the open field test. At 6 months old, we start validating their learning and memory deficit using the novel object recognition test. And we repeat all those tests by 9 months old time point. After finishing all the behavior tests, let's see some data. At 3 months old time point, as shown here, we didn't see much of the motor deficit in the HUGO strings. As indicated here, there were no difference detected in between any of the HUGO line in comparison to the black 6. Also, regarding their anxiety level, we didn't see any alterations. As shown here, there are no differences. The rotor-road result corroborates our findings in open field test. We haven't seen any motor deficit in any of those lines, which are also as expected because 3 months old is too early for any pathological changes. Things start changing at 6 months old time point. Neither the motor function nor anxiety level changes, but we did see some trends without any statistical significance. More interestingly, we have observed a clear cognitive impairment in P301S strain using the novel object recognition test. Here you see the comparison or the time spent for the mouse to observe, to explore the familiar object in comparison to a newly put object in the arena. Based on the mouse nature, they are more interested in exploring the new object as long as they could recognize it. So you see that the black 6 mouse, without surprising, can recognize the null object. The HUGO wild type can also do that. P01S, P01L, yes. However, not P01S. They sort of lost the ability to recognize the novel object and get mixed with them with the old familiar object. That being said, by 6 months old, in the HUGO P01S strain, there are observable cognitive impairment, which is also correlate with human diseases because human patients with P01S, they do have an accelerated pathophysiology. Things get even more clear by 9 months old time point. Here we see both the P01L and P01S strains showing cognitive impairment in the null object recognition test. That being said, though a little bit late, however, P01L mutation as the P01S also generate cognitive impairment. Here is a small summary. Across all 4 strains, we didn't see any pathology in the black 6. Naturally, we also didn't see any pathology in the HUGO wild type, meaning the human MAPT gene works properly in the mouse. However, we did see a pathology in the P01L mutation by 9 months old, indicating this is a late onset, slowly developed cognitive impairment. And we see a cognitive impairment in the P01S strain by 6 months old, indicating this is a relatively early onset and accelerated pathology. Once we have these behavior observations, we continued with pathological and biochemical studies. Using the brain tissues, we try to observe the tau tangles in the mouse brain as indicated here. To this end, we utilized 3 different antibodies, focusing on developing human tau in our HUGO lines. We used HT7 for total human tau. Based on HT7, we also used the AT8. Now that we have gathered some information from the HT7 staining and also the Tau5 biochemical detection validating that there are indeed exclusively human tau exist in the HUGO mouse strain, but not the black 6, we continued to validate the existence of pathological tau, namely AT8. As shown here, we could not detect AT8 in the black 6 brain as expected. We also didn't detect the AT8 in the HUGO wild type, also as expected because there were no pathological behavior in those strains. But we detected strong aggregated AT8 signal in the brains of P01L and P01S, corroborating their behavior pathology. To summarize this, on the behavior side, we didn't see any pathology in black 6 and HUGO wild type, indicating the normal functioning of humanized MAPT gene in the mouse. We detected pathology on the behavior side in the P01L and the P01S mutation at the respective time point, corroborating their pathological advance in human patients. On the pathological side, we detected murine of human tau in corresponding strains, but only human tau in HUGO strains, not in the black 6. We detected 88 phosphorylated pathological tau only in the P31L and the P31S strains, but not in the other two strains, as expected. That being said, in conclusion, our HUGO mouse strains carrying human MAPT behave as expected as their human counterparts in tau-related diseases. Now that we have a rough understanding of our HUGO mouse strains, we should discuss why we are interested in this. To start with, we could use Alzheimer's disease as a good example, since it has both amyloid and tau pathology simultaneously. Here are two demonstrations from literatures. As you can see here, the AD tau pad is here, while the AD amyloid pad is here. Also, there is another AD tau pad. So, that is but one demonstration that tau pathology as a pathological marker has a higher clarity in comparison to our generally used AD markers like amyloid PET. It is more precise, it is more clear. In addition to this, more than one clinical studies, have demonstrated that in comparison to A beta, tau has a more clear correlation with cognitive impairment. That being said, with cognition declines in 80 patients, tau as a biomarker can more clearly reflect this. As you can see here, regardless for the phosphorylated tau 217 or tau 181, the p-value is much more smaller in comparison to the inconclusive A beta 42:40 ratio. That being said, with the disease progressing, tau as a biomarker can more clearly reflect its process. Being said that, tau is not only a good biomarker for evaluating the disease progression, it is also a valuable target for intercepting the disease progression. Here are demonstrations about the ideas summoned in recent years to intercept tau pathology in the cells and in the disease. And here are two news from the industrial side that the Voyager and also another company developed a drug specifically for tau immunotherapy. Actually, those clearly are not only therapies for intercepting tau. Until 2025, there are more than a dozen new therapeutic strategies for intercepting tau, including not only the immunotherapy, but also therapies from other aspects, something like the aggregation inhibitor or microtubule stabilizer. These are all therapies focusing on tau, not only to diminish tau in general or pathological tau, but also to stabilize it, to prevent it from aggregating, to prevent it from transforming itself from a physiological part to pathological part. It should be noted always that tau is a protein with clear physiological functions. Hence, treatment for tau cannot be just diminishing tau, which is dangerous and compromising for normally physiologically functioning human body. However, targeting only pathological tau is an extremely difficult task. This is from literature composed in 2023, that is 2 years ago. After 2 years, only very few treatment continues to the phase 1 or phase 2 trial, and others just diminished in the process. Based on this, we generate an idea that we believe intercepting tau in any tau-related disease may not be a task that can be achieved by targeting only tau. We think there are plenty of opportunities to intercepting the entire tau pathology using not only tau as a target, but also other tau-related processes, such as here indicated the acetylation inhibitor, the antisense oligonucleotide, and also source of inhibitors and stabilizers. Based on this, we would like to explore further, in addition to our current HUGO humanized MAPT models. We call it Human MAPT Plus. That means this mouse model will carry not only one humanized gene as the MAPT, but also other humanized gene related to potential tau-associated therapeutic strategies. With this, we could not only simulate tau pathology in one mouse model, we could also give opportunities for researchers, for scientists from both academic and industrial side to explore new therapeutic translational strategies for these diseases. Not only for the humanized MAPT, we are also developing humanized APP+, humanized SNCA+, humanized TERDBP+, for diseases such as Alzheimer's, such as Parkinson's, such as ALS. We offer more comprehensive humanized models for not only studying the disease pathophysiology, but also exploring more translational, diagnostic, and therapeutic strategies. That is my share for today, and I look forward to hearing back from you all.
Thank you, Dr. Cui, and for that insightful presentation. So the findings you shared are truly groundbreaking and open up exciting avenues for AD research. And now we'd like to open the floor to our audience. And we have received a number of excellent questions throughout the presentation. So let's begin the Q&A session for the ones that we collected. And the first question is, well, they actually have 3 parts. With the HUGO GT tau mouse model, could you walk through how it really mimic human tau pathology, especially for the 3R and 4R ratio? And for the NFL and neuron loss. And the last one is, have we seen 6 dimorphism in this model?
I think we should go through the questions one by one. That will make it much easier.
Sure.
Okay.
Do you want me to repeat it or?
Let's go with the first question.
Okay.
I kind of heard that how this model simulate the human tau in the 3R and 4R ratio, right?
Yes.
Yes. Regarding the 3R, 4R tau repeat, as you just mentioned, we observed a different expression pattern in the tau mouse models. As you can see here, using the tau5 antibodies, we could see different bands in the BLAC6 and also in the HUGO lines. However, as we currently developing a new method to enlarge this section and to see if we can clearly see all the tau isoforms, we will be in the future several weeks to answer this question in a more precise way. But regarding how that 3R, 4R ratio actually impacts human tau-related disease, based on the knowledge in the current knowledge in the field, there are no clear theory. We In a way, we believe that the hyperphosphorylation targets really impact on the disease, but the 3R/4R ratio, how that impact on the disease, is open for explorations. Also, since we have observed, as shown here, 88 and also the aggregates of tau, hyperphosphorylated tau within the neurons, without clear statistic, however, we did observe some neuronal loss in this area. By the way, this is the hippocampus, the center of memory, and we observed in this P301L and P301S, the hippocampal region, the neuron from hippocampal region, they become more disorganized, they become more differentially shaped, some of enlarged, some of shrinked. That means extremely unhealthy states of all those neurons, which usually leads to neuronal loss, if that would answer your question. By the end of that, I believe you asked something about sex dimorphism or sex difference in this model. Well, that's a very good question. Since we did observe some difference in A beta-related AD models. However, in terms of tau-related AD models, there are currently no observable sex difference. I hope that answers your question. And frankly speaking, I do need some help regarding the second one. I cannot memorize that. So what was the second question?
Is the NFL and neuron loss. So
well, the NFL stands for the neuron filament, the neuronal filament tangles. So as you can see here, we see some aggregates, small aggregates within the N, the neuron stained region. And we believe that is the neurofilamentary tangles. If we have to further study that, it requires confocal microscope or even electron microscope that is beyond our current scope, meaning validating the model. So as far as I can say, I can tell you, I can show you that there are aggregates within the neurons. The neurofilament tangle is something I cannot 100% give you an answer, scientifically, prudently speaking.
Thank you, Maurice. That's a pretty clear statement. So the next question is, how is the disease progression and pathology compared to the existing tau models? And are we anticipating any locomotor dysfunctions, which doesn't manifest in 9 months?
Frankly speaking, that is quite difficult to answer because the current most prevalently used tau model is called PS19, aka P3S model. This is a transgenic model, and like all transgenic models, the transgenesis itself is a random process. So the insertion, the transgenesis, the dosage can be different from colony to colony, from generation to generation. But roughly speaking, the disease manifestation of this line is somewhere between, the memory and learning deficit disease manifestation starts from somewhere 6 to 7 months, approximate to our observation in our P301S HUGO strain. However, frankly speaking, we didn't observe the motor dysfunction in this line, which was observed somewhere early in 2 or 3 months old. But one thing should be noted. We didn't, during our process of literature review, see the motor deficit be observed in all the literatures, which is also corroborating that the transgenic mouse lines sometimes can be unstable. That would be my answer.
Sure. And the next question is, do we have plans to cross the tau mouse model with other AD models like humanized APP or TFRC?
That is a very, very good question. As we all know that the immune therapy, the passive immune therapy infusion, the human patients with anti-A beta, anti-tau antibodies, are the current most advanced and validated therapy for Alzheimer's disease. And in order to transfer a big protein like antibodies from the peripheral system to the central nervous system, we have to pass blood-brain barrier. And the TFRC or the TFR1 receptor is currently the most validated receptor. Hence, of course, as I just mentioned, our mission here is to supply the industrial and academic side the more validated and usable disease model for disease pathophysiology and for therapeutic strategies. Yes, we are currently crossing all our tau strains with the humanized TFR1 strain, making it suitable for developing any kinds of antibodies against either tau or A beta.
Nice. So we are expecting more valuable models.
Yes, in the future several months, it still takes time.
Yeah. And then we have two more questions. As Cyagen offers CRO services, can you give a real-world example, like a quick case study that shows how it truly simplifies the AD research workflow?
That is a tricky question because a real-world example would be a project which we conduct with our clients, and usually that kind of thing is confidential. However, we did conducted some internal studies to see if our model actually works with the real-world example. So we acquired some ASO, the antisense oligonucleotide, against tau sequence to see if that actually works with our models. We did that because the ASO is specially designed for human but not murine tau sequence. So if that works, it works only with HUGO models. And we have tested that. We have conducted several weeks treatment and extracted their brains, their blood for further studies using the ddPCR and also qPCR. And as expected, the ASO didn't work very well. It has some effect, of course, with the Black 6, but it works extremely well with our HUGO mouse strains, indicating it can diminish tau generation in HUGO mouse lines, but not in the Black 6, indicating both the ASO is designed for human and the HUGO strains contains human sequences. Of course, this is a very preliminary studies, and as I just mentioned before, lowering general tau concentration does not really help disease recovery, since tau is a normally required protein. But I believe our future clients may have a better drug for this kind of thing. And that also indicates how fast our model can generate a solid result for your question, for you to be tested therapeutic strategies.
Sure, that's pretty comprehensive. And here we go, the last questions. Did you get any early feedback or interesting questions from your customers who use this model? And did their input end up influencing the validation process that you just described?
Well, our validation process is why we can sell these mouse models to our clients. And in terms of the feedbacks from our clients, the most direct feedbacks is we are getting more and more projects from our clients, regardless old and new, inquiring and requiring these specific humanized tau models for more projects. So I guess they are pretty good for their project, internally or externally speaking.
Sure, very insightful. So we are embracing helpful feedback from our customers, and the collaboration is also welcomed. And of course, a huge thank you to Dr. Cui for sharing his expertise with us today.
Thank you for inviting me. I surely look forward to sharing more interesting information with all of you in the future several months.
Before we conclude, I'd like to share some exciting updates from Cyagen. For our academic clients, we have two significant programs currently running. The first one is the buy one get one offer on catalog knockout mouse models, which is an excellent opportunity to expand your research capabilities. And the second one is the Cyagen Trailblazer Grant Award, CTGA, where we're providing over $1 million in total funding to support groundbreaking research projects. And in addition to our rodent model services, Cyagen also offers a range of cell-based solutions. We are currently running a 50% off on iPSC editing services for our new customers. For more information on any of these campaigns, our services, or to discuss your specific project needs, please visit our official websites at www.cyagen.com. And we will also provide the link in the below section if you're watching the replay. And you can also follow our official LinkedIn account to stay up to date on our latest news and events. And on behalf of our entire team at Cyagen, thank you again for participation, and we hope to see you at our next events. And have a wonderful day. Thank you, Maurice.
Thank you very much. Ciao.